All right, on this side we'll talk about some health issues, more in the health and the medicine area of aldehydes and ketones. And one of them is formaldehyde. It's the simplest aldehyde. It's actually used in a lot of resins and plastics. Sometimes when you buy a new car, you kind of have that new car smell. A lot of times it is formaldehyde because it's used in order to make the plastic and all the coatings on like the seats and the plastic and stuff like that. Formalin is actually a mixture of formaldehyde and water, and that's what's used to preserve animals. So if you go into like a cadaver lab and there's that classic smell, that's the smell of formaldehyde. Acetone is also used to make materials and polymers. It's also used as a solvent. We obviously use it in lab to clean our glassware. All right. It is also produced in the breakdown of fatty acids. Okay. You might have heard if someone is a diabetic, they start spilling ketones into their urine. Okay, they're in ketosis. Also, there's a diet called the ketogenic diet. When your body cannot break down glucose anymore because you have a lack of insulin, your body then has to find an energy source other than glucose. Because if you no longer have insulin, your body cannot use glucose as an energy source. So it starts using the fat in your body as an energy source. And when your fat breaks down, it breaks down into ketones. Okay, and acetone is one of them. So when you go into a state of ketosis, that's because your body can't break down sugar anymore. If you're on a ketogenic diet, what technically is considered a ketogenic diet is when you are consuming less than 5% of your diet has carbohydrate in it. So a ketogenic diet is where you have 60 to 70% of your diet is from fat. The additional 20 to 25% is from protein, and then you're eating less than 5% carbohydrates. So you're basically starving your body of carbohydrates. So the only source your body has of energy is to break down fat, and that's what they say is the ketogenic diet, is that it burns the most fat because that's what it primarily uses as your energy source. And if you are on that diet, you kind of have to check your urine every day just to make sure that you're in a state of ketosis. But if you're diabetic, you no longer have insulin, and your pancreas so your pancreas does not produce insulin, and your liver can't break down any glucose. So the glucose just kind of runs through you. So your body has to use energy somehow. So it starts breaking down all the fat in your body, and it turns it into acetone. So you can see unusual high levels are found in a diabetic patient. Some kind of common aldehydes and ketones. This is cinnamaldehyde. All right, you can kind of see it is basically the main compound in cinnamon. All right, you can see the aldehyde right here. Vanillin is another one. You can see that you have an aldehyde right here. It's an aromatic ring, just like cinnamaldehyde, and you have an aldehyde. Okay, citral and citronella. Everyone has heard of citronella candles. These are aldehydes as well. These are very similar in structures. There's a little bit of variation of a single bond here and a double bond here, a double bond right there. This compound you find in citrus fruits as well as in the lemongrass. It's used in the perfume industry, which has a very nice smell to it. It's also used in the synthesis of vitamin A, which is what our eyeballs use in order to see. Citronella is used as a mosquito repellent. This also comes from citrus fruits, which kind of keep the whole point of it is the fruit is protected from insects because of that smell. Okay, We'll continue. That was a kind of a quick section. This is kind of reactions now with aldehydes and ketones. So if you have an aldehyde, what kind of chemistry can you do to it? Well, you can oxidize it up to the carboxylic acid. I believe we talked about this in the alcohol chapter. You can take an alcohol and oxidize it to a carboxylic acid with an oxidizing agent. And if you remember, the oxidizing agent was potassium dichromate. That was the very common oxidizing agent. So you can take an aldehyde and oxidize it to a carboxylic acid. You can also do what's called a reduction, where they show this hydrogen here, which means I'm going the opposite way. Rather than oxidizing it, I'm getting rid of the double bond and I'm turning it pretty much into an alcohol. So a lot of times this will be hydrogen, which we will see an example. So one hydrogen will go here and another hydrogen will go here. So you can see you can take a ketone and you can turn it into an alcohol. Okay, and I believe the common reagent that your book uses is hydrogen. You just use hydrogen. Okay, so in the oxidation reaction, you convert pretty much the oxidizing, the H becomes an OH. You basically add an oxygen in there. Okay, and we said that the potassium dichromate is the common oxidizing agent. Now, if you have a ketone, however, you don't get a reaction. Notice you have to have a hydrogen here. You need it. So an aldehyde will give you carboxylic acid. The ketone gives you nothing. Okay, you need a hydrogen on one of the sides. Okay, another oxidizing agent your book talks about is this silver oxide and ammonium hydroxide. It's called the Tollens reagent. It's a very common reaction, these compounds selectively oxidize aldehydes, because notice you have an alcohol you can also oxidize. But by using the Tollens reagent, you just oxidize the aldehyde group. You don't oxidize the alcohol. If you want to oxidize everything, then you use that potassium dichromate. That will oxidize everything to the ketone and the carboxylic acid. Okay, so reduction is the opposite of oxidation, which I just talked about. You basically are adding hydrogen in place of it to form an alcohol. So the common reaction is hydrogen gas, or hydrogenation. So for aldehydes, you get primary alcohols. For ketones, you'll get a secondary alcohol. All right, so H2 gas in the presence of a palladium catalyst is a common reagent. So this is kind of a reaction that you might want to add onto your card for exam 2. All right, so ketone plus some sort of reducing agent, which we said is hydrogen on a palladium catalyst. That's kind of a line in separating hydrogen and palladium catalyst. You get a secondary alcohol. Okay, so here's kind of a summary. If you take an aldehyde plus hydrogen, you get an alcohol. If you take a ketone plus hydrogen, you get a secondary alcohol. Pretty straightforward. All right, this is just to kind of show you how this reaction is used in the synthesis of Prozac. All right, here's the structure of Prozac down here. All right, this is kind of what they're starting with. That's their starting material. And they hydrogenate off that carbonyl to form the alcohol. All right, this group right here is equivalent to this group right here. Okay, so these two carbons are these two carbons, just to kind of show you a real-life example of this, when it's used and where it's used. It's a very common reaction. All right, in our bodies, we do not have hydrogen gas, okay, but our bodies are constantly doing oxidations and reductions all the time, but we do not have hydrogen and palladium inside our body. That would be pretty darn bad, okay? So what does our body use as a reducing agent? Our body uses the coenzyme NADH. For those of you that have taken a bio course or a micro course or A&P, you've probably talked about NAD and NADH. Okay, that is our reducing agent. In fact, this hydrogen on the NADH is the hydrogen that goes on there. So instead of hydrogen gas, we have this hydrogen on the NAD. Okay, so you add NAD. I believe you need like 2 equivalents of NAD, or I'm sorry, no, the other So this hydrogen right here is coming from NADH. Okay, this hydrogen is coming from acid that we have in our body. We always have very small amounts of acid on our proteins and our waters. We're talking tiny, tiny amounts of it, but it's enough in order to put that proton on there. Okay, so basically this hydrogen is coming in here, all right, adding it to there, kicking up these pi bonds, and then eventually a hydrogen off of the enzyme is going onto there. All right, so we don't have hydrogen, but the H in NADH is our reducing agent. Okay, so in acting as a reducing agent, the NADH is oxidized to NAD+, which is a biological oxidizing agent. This now can go ahead and do an oxidation somewhere and pluck off another hydrogen to reform NADH. So that's basically what NADH and NAD+ are doing in our bodies. It first does a reduction to NAD+. Okay, so it's reducing it. Remember, reducing has an H. Then this will go into our bodies and do an oxidation and regenerate. And that's basically what it's doing. It's going back and forth, reducing, oxidizing. So it reduces one reaction, then it goes back into the cells, and it does an oxidation reaction of something else, and it just keeps going back and forth. Okay? Why don't we stop right here, and the next video we'll start up with 16.6.